You're sitting at home, scrolling through your phone, when up pops a notification from your favorite social media app. You have a new suggested friend. But wait, how did this social media app know to suggest this friend to you? The answer? Networks. It's all about who you know, who your friends know, and so on. The truth is that networks have been used by scientists for decades for understanding social ties, traffic patterns, and brain chemistry. Well, it's even true for microbiomes, the collection of microorganisms, bacteria, fungi, viruses that live in, on, and around us. For example, scientists estimate that soils contain 1 billion microorganisms per gram, which could represent millions of species. And within these webs of diversity could be trillions of interactions. But why should you care about these microscopic social networks? Well, it turns out that these relationships aren't tied to how a microbiome functions. At Berkeley Lab, we're interested in understanding the links between plants and their microbiome, as well as between microbes within the microbiome. And this is because plants, just like us, form symbiotic relationships with microbes to help them grow better, especially under extreme environmental stress like drought. But how do you survey a microbiome? With its DNA, of course. It tells you who's there, in what abundance, and with whom else. And so for a while, we knew that stress could change the diversity of microbes that plants associate with. With network science, I was able to discover that it also completely reorganizes the network. In fact, it changes not only who associates with whom, but also who serves as hubs, which you could think of as the social butterflies, the most highly connected taxa that serve as keystones in the network. I also found that these microbes become more connected, especially among taxa believed to help plants grow better. And most surprising, while we thought that stress would increase competition for resources, we actually found that it might promote cooperation and aid in microbiome stability over time. So, this is only just the beginning, because each microbe in the network has its own story to tell and its own genome filled with information to uncover. The next frontier, linking these relationships to their genomic and metabolomic mechanisms. With this, we'd be able to better suggest microbes to use in biofertilizers that could help plants grow under a rapidly changing climate. With this, we might even call it Facebook of the field.